just to introduce Priya a little bit, I think some people that were attending the Southern Alberta Beekeepers meeting last year had the pleasure to meet Priya and to hear her talk. She was here in Alberta last year to talk a little bit about bee nutrition. And so Priya is a research associate at the University of Oregon State University.
Oregon State.
And thanks, Priya. And Priya does her research mostly focus on bee nutrition and everything that with the proteins, amino acids, and as I think she'll be talking about the sterols, right, Priya?
Yes, yes.
So, which is very interesting, and I think increases a little bit of our understanding about bee nutrition and how we can supplement when needed to the best possible we can, close to nature and what they naturally need. So Priya, if you want to start your talk, I will mute myself and turn off, and then I'll come back at the end.
Thank you so much, Renata. Good afternoon, everyone. Thank you so much for joining me here today. And thank you, Renata, for extending the invitation. It's almost a year and it's lovely to be back amidst you, even though this year it's virtually. So those of you who have heard me speak last year, as Renata mentioned, I do work on bee nutrition. I work on impacts of pesticides on bees. And I also work at the interface of how these multiple stressors, they can be interrelated to each other. How can they work in synergy with each other, affecting bees? But today, my talk is all going to be on bee nutrition. So we know that there are multiple factors which are causing bee declines. For example, the constant stress of transportation between the different commercial crops or moving the colonies around, poor nutrition for habitat loss, suboptimal nutritional supplements, or even monocropping. Then we have pests, parasites, pathogens, environmental stressors, a lack in genetic diversity of the stocks, as well as, of course, the impacts of pesticides. With all of these factors either working alone or working in synergy with each other, it is important that we understand how nutrition can play an important role. Because if your hive is well fed, if your bees are healthy, if your bees are strong, they can withstand other forms of stressors. That is why we often call nutrition the first line of defense for a colony. So there are many ways a honeybee colony can face nutritional challenges. There can be a lack of pollen abundance. The quantity of pollen that is coming into the hive is not enough. There can also be a lack of pollen diversity. Too much of only one type of pollen is coming in. There is not much diversity. There can also be a lack of pollen quality. For example, what we see here in Oregon for blueberries or hybrid carrot seeds, these pollens, their quality is not as good as something, say, for example, almonds. So even though the bees are bringing back in some pollen, that pollen is not of a high nutritional quality. There can also be insufficient nectar flow, as well as, of course, as beekeepers, we have to constantly keep an eye on how well these bees are going to be able to survive the winter. So the diatoneous bees, the bees that start out in fall, survive through the winter and raise the next generation of brood in spring. These bees are very important. They are the longest surviving bees. And of course, we need to make sure that these bees are well fed and healthy at the onset of winter so that they can raise the next generation of bees in the following spring. When we think about honeybee nutrition, there are two major components. We have macronutrients, which are, of course, as the name suggests, they are extremely critical for bees, and they are also required in larger quantities. Examples include carbohydrates, proteins, and major lipids. We also have micronutrients. They are equally important for bees, even though they are required in smaller amounts. And the examples include phytochemicals obtained both from nectar and pollen, vitamins, which mostly come in from pollen, minerals, mostly from pollen, as well as phytosterols. Pollen is the only natural source of phytosterols for bees. And my talk today will focus a little bit on phytosterols and briefly touch upon the other points. So after discussing with Renata, we decided that today I'm going to stick to only a couple of relevant publications, 2 or 3 publications which are recently published from our lab. So the first publication that I will be discussing is this particular publication. It is open access. You can freely download it, and I'll be happy to share the link with anyone who's interested later on. The second publication is another journal called Molecules. This is also open access and it's free to download. And the third publication, all of these three are sort of like the story is knit together. So this publication is also open access and free to download and is in a journal called Metabolomics. We know that bees are, of course, they're important in not only pollinating our crops, but they also generate revenue. If we just look at the almond pollination in California, it generates about $11 billion annually in revenue. However, to be able to pollinate these almonds, these bees are forced to start their brood cycle earlier. Beekeepers all over the US, and I'm sure even in Canada, all across North America, beekeepers are concerned about uh, about improving bee health, and we understand the need for providing better nutrition to the bees. Studies have shown that colonies which are only provided these commercial crude protein diets alone, these colonies, they do not perform as well as those colonies which have access to abundance of natural pollen. In fact, when we think about bee nutrition, the major focus for decades in studying bee nutrition has focused on the macronutrients, which mostly include proteins and carbohydrates. But research on micronutrients, this is severely lacking. So important micronutrients such as phytosterols, phytochemicals, vitamins, what's happening with these? This particular area of research, it is still underappreciated. So why do we study sterols? This is because all insects, including bees, they are sterol auxotrophs. This essentially means that they cannot synthesize their own sterols and they need to source these sterols from an external diet. Sterols are very important because they are precursors for important insect hormones such as molting hormones. They are also components of insect cellular membranes. So they help maintain the structural integrity of the insect cellular membrane. All insects have a different sterol need. All bees, they also need different types of sterols. So if we just talk about honeybees, they in particularly, they require this sterol called 24-methylene cholesterol or 24-MC. So this is the only sterol that honeybees can effectively utilize and metabolize. Let me show you this quick animation to sort of explain better how this particular sterol is important for honeybees. So as I mentioned before, pollen naturally has an abundance of phytosterols. So when a nurse bee, she consumes pollen, she then selectively takes up these sterols into her body tissues as endogenous sterols. These endogenous sterols are then selectively transferred to the growing larva through brood food. Thereby, the larvae have sterols, they have molting hormones, and successive generations of bees are being produced. Now, for example, if these artificial protein supplements, they lack these critical phytosterols that honeybees require, after a point of time, there will be no new sterols to replace these endogenous tissue sterols in nurse bees. No new sterols going into the larva, no molting hormones, and after a point of time, no new generations of bees. So this is why these phytosterols are extremely critical for honeybees. in fact, for all bees. So, to understand how these sterols can be important, we decided to first look into the literature because we know that, okay, there's this one study which has told us that of all the sterols, 24 methylene cholesterol is the sterol that honeybees need. But how much do bees need? Is there a percentage of dry diet that needs to contain the sterols? How can the sterol benefit bees physiologically as well as in terms of their colony health? So, to answer all these questions, when we started digging into literature, we realized that the last few studies that were conducted were early 1980s, late 1970s by a group led by Herbert et al. We understood that there has been decades of gap in research, decades of gap in understanding as to how these sterols can be beneficial for honeybees and how much of these sterols are required. So, to know more, we conducted this study in the lab. What you see here are lab cages where we have emerged nurse bees and we have released them in these lab cages. There are about 200 honeybees in each of these lab cages. And we gave them sugar syrup and water on the top and an artificial diet at the bottom on this petri dish. So these artificial diets, they contained equal amounts of amino acids, vitamins, minerals, salts, carbohydrates. The only thing that varied between the diets was how much of 24 methylene cholesterol we added to it. So we had, of course, a control which had no sterol. Then we gradually increased the amount, the concentration of sterol in the diet, all the way from 0.1% to 1.0%. And we conducted this study for 3 weeks. And we wanted to see at which concentration of the sterol we could see the maximum benefit, if there was any, in these honeybees. We found that the survival in the bees was significantly higher when they were given higher sterols in their diets. We also found that an important physiological marker, such as the het protein content in these bees at the end of 3 weeks, was significantly higher in the groups which consumed higher sterol diets. In fact, their consumption was also more. So the bees which had more sterols in the diets consumed the diet more than bees which had less sterols. Abdominal lipids, so the lipid content in the honeybee abdomens, they were also significantly more when they consumed more sterols in the diets. We also found that the lipids in the honeybee head capsules, they were also significantly higher when more sterols were added to their diets. So with this, what we understood is that the more the sterol in the diet, their consumption increased, their survival was better, and important physiological markers were also better. And most of these changes we found from a diet of 0.5% of sterol in the artificial diet. So this told us that 0.5% is certainly a good number to start with by adding it to the diets to help improve bee health in terms of sterol nutrition. So to understand more as to how this particular sterol can also influence the overall colony health as well as colony structure, the queen health and all. So now we are expanding the study into these semi-field scenarios where we have constructed these flight cages. We have released a nucleus hive in each of these flight cages. We are giving them these artificial diets with different concentrations of 24 methylene cholesterol. And we are now going to track the uh, health of the colony over a period of 8 weeks. Another thing that we wanted to know is that, of course, so we are now seeing that this particular sterol, it is improving bee physiology, it is improving bee survival. So it definitely has some benefit on the bees. But what we are also interested in asking is that, so if when we feed the sterol to a young nurse bee, how does the sterol move across the bee tissue? Is it more in the head? Is it more in the abdomen? Is it more in the thorax? So to know that, This, what you see here, is what typically a molecule of 24-methylene cholesterol looks like. And this particular carbon right here, we replaced this carbon with a stable isotope of 13C. Imagine this to be your beacon in the dark. So suppose you are walking through a dark tunnel and you have a flashlight in hand, and if you switch it on, I, looking at you from the outside, I know exactly where you are and exactly how you're moving because I can see your flashlight. So this molecule right here, this carbon right here is the flashlight for this entire molecule. When we quantify this carbon isotope from the honeybee tissue, this will tell us where this molecule has traveled, how it has traveled, and which tissue it has accumulated more in. So to understand that, we conducted another similar study where we released these newly merged bees in lab cages, gave them the artificial diets, and we found that their survival, their consumption, head protein, and abdominal lipid was higher in the group which was given this particular sterol in the diet. So let's see what we found. How was the sterol accumulated in the honeybee tissues? I know that this graph looks very complicated, but let me walk you through it. And I'll always be happy to come back to this slide if you have questions for me later on. As you see, each of my slides are numbered at the bottom right. So here, if you follow my laser pointer here, these bars right here are total carbon. So the first four bars show you total carbons in the head region of the honeybees. The next four bars show you the total carbon in the thorax of the honeybees. And the last four bars show you the total carbon in the abdomen of the honeybees. W1 indicates week 1 because we conducted the study for 4 weeks. So at the end of every week, we collected live honeybees. We separated out the head, the thorax, and the abdomen, and then we quantified the carbon isotope and the total carbon. And if you remember what I mentioned before, the carbon isotope tells us how much of that sterol is present in that tissue. So what you see here, W1 indicates week 1, W2 week 2, W3 week 3, and W4 week 4. H for head, T for thorax, and A for abdomen. The blue line right here, what you see, is that percentage 13C. That is the isotope that we had quantified. So if you're just looking at it, week 1 head, week 2 head, week 3 head, week 4 head, you'll see that week 1 head had the highest accumulation of this sterol compared to weeks 2, 3, and 4, where the number slightly decreases. If you look at thorax, even though if you look at the blue bar, it slightly increases at the end of week 4, there was not much of a difference. But if you look at the abdomen, look at the blue bar. Here is week 1 through week 2 through week 3, and look at the sharp increase between weeks 3 and 4. This tells us that more of this was accumulated in the honeybee abdomens during weeks 3 and 4. So what did we find? What we found was interesting because this was comparable with the honeybee life history traits. As a young nurse bee, her primary responsibility is to be able to feed the brood by producing more of the brood food, be it worker jelly or royal jelly. And as sterols are passed on to the brood through the brood food, she certainly will accumulate more of the sterols in her head regions when she's still conducting the nursing functions. As she grows older, she has switched from her nursing duty to a forager duty by the end of, say, weeks 3 to weeks 4, when she is at least 21 days or older. So now all she has to do is she has to store enough of this sterol in her abdomen so that she can revert to a nursing responsibility if the needs be. Otherwise, have just enough resources in her body to be able to fly out, forage for food, and bring it back. So this is what we found. And maybe that is why there was not much change in the thorax, but the head and the abdomen showed the more drastic changes in how much of the sterol was accumulated over time. So this certainly tells us that 4 weeks may be a good amount of time for which a honeybee can go without taking in sterols, after which these phytosterols, 24-methylene cholesterol, has to be supplemented to the colony so that the nurse bee successive generations, they can continue to perform their functions. With that, the third question we had in mind, if I can reiterate, the first question was how much of this sterol is needed and how can it be beneficial? The second question was how is the sterol moving across the honeybee over time and how can it help improve the colony health? The third question that we have is that now that we know how much of the sterol is needed and which sterol is important, how is it benefiting honeybees? How do we know which plant has how much of which sterol? Do we know the sterol profile of the different pollen sources that are available for bees? So to know that, this is another area that we are currently working in to understand the need for better forage. The habitat for bees, it is mostly chosen based on relative attractiveness. It is not based on the nutritional quality of the plant itself. Do we really know that this particular bee, be it a bumblebee or a honeybee, if it's attracted to a particular flower, is it because that flower is the closest to its colony? Or is it because that flower smells good, it looks good, or is it because that flower is actually providing a good nutrition to that particular bee? No such information on the micronutrient profiles are available for plants in North America, and there is also a very limited information available for proteins and lipids. Thus, it is very important that we understand the nutritional profile of these commercially pollinated crops as well as the non-crop forage, and also understand the nutritional quality of the commercially available dietary supplements that we provide to the bees. And with this, our lab, we want to be able to build a database for pollen nutrition, which can be available to all bees, be native bees or honeybees. With this will be able to provide feasible practical recommendations for not only beekeepers, but also for growers. So if you're a beekeeper and you know this is what your bee has, has access to, if you know what those pollen can provide, you can either plant different forage around it to complement the other nutrients that the bees need, or you can mix things to the commercial diet, or you can make sure that you're storing pollen during times of pollen flow and providing it to your colonies in times of pollen dearth. So we were very recently awarded with a USDA grant to exactly do this. We got a grant to build the bee nutrition database. And with that, we want to now look into the pollen protein content, pollen lipid content, nectar sugar percentage content, pollen phytosterol content, as well as the pollen metabolites and phytochemical contents. We have already actually started working on this, and we have a big team here at Oregon State. You can see my PI, Dr. Ramesh Sagili, right here. And I have two other colleagues and collaborators who work with us at the Mass Spectrometry Center at Oregon State. This is Jeff Murray, and this is Dr. Lipinyan. So we have been constantly testing many different types of bee diet sources, which include some commercial pollen sources that we found, for example, apples, plums, cherries, almonds, and apricots. We are also testing some vegetable oils that beekeepers, at least around here, they tend to mix in their patties, such as borage oil, canola oil, and soybean oil. We are also trapping for pollen. We use our experimental hives in some of these orchards, and then we trap for pollen. So we've collected pollen from pears, almonds, and blueberries. And we've also tested some commercial diets as well as, of course, honeybee tissues to know exactly what sterols are they assimilating. So I just wanted to highlight that our lab is probably the only lab which can exactly quantify the amount of phytosterols that is present in the pollen as well as in the bee tissues in parts per million. This took us about 2 years to standardize the methods, and we can now test about anywhere between 12 to 14 different types of phytosterols. This is important because, as I mentioned before, each bee species has a different sterol need. So, what bumblebees need will not be what honeybees need, will not be what mason bees need. So, this way, if we are able to know a big range of the different types of phytosterols that are present in these plant pollens, we can, of course, provide recommendations for not only honeybees but also for native bees. So if you just look at these two pie charts, each color represents a different sterol. Even if you don't read anything, to your left is canola oil, to your right is the commercial dye. If you just look at this right here where my cursor is, this is 24 methylene cholesterol. It is a tiny slice right here of the pie, but we hardly found it in the commercial diet that we tested. Look at this narrow, thin line where I have my cursor moving. So if you just look at the color differences, you'll realize that why it is important to build this database, because each diet source has a different proportion of the phytosterols. Right here, what you see are to your left, right here is commercial almond pollen that we collected, pure almond pollen. And this is corbicular almond pollen that we trapped from the honeybee colonies. So they are more or less quite similar in their, I would say, structure of the different phytosterols, the composition of the phytosterols. And if you look at 24 methylene cholesterol, look how much of it is present. It is in fact the majority of the phytosterol in this particular almond pollen. So this certainly tells us that how rich almond is for honeybees. The same way, each bar here shows you a different diet group. It, this is borage oil, canola oil, soybean oil, commercial diet, and different types of pollens. Each color for every bar is a different phytosterol. If you just don't worry about reading anything, if you just look at the range of colors, you can see how different each diet source is. And plant pollen, naturally, they have so much of 24 methylene cholesterol that is absent in the commercial diet and somewhat present in the borage oil. Similarly, we are also looking into metabolites, where we are looking into the different phytochemicals, we are looking into amino acids, different flavonoids. So same as before, the two pie charts, they show you to your left, which is a corbicular almond pollen, to your right is a commercial dye. This is just a very simple example of amino acids. Each color is a different amino acid. If you just look at the colors, look how different they are. So you can make out how different the ratios, the proportions of these amino acids are. Same with borage oil and honeybee tissues. Look how different they are if you are just looking at the colors. So each diet source certainly has differences in the proportions of amino acids and other metabolites that they offer for honeybees and other bees. Same as before, each bar is a different diet source. Each color is a different amino acid. If you just look at the relative abundances, just look at their proportions, you'll see that what a difference there is between the vegetable oils, the commercial diet, and the natural pollens. The commercial diet certainly has good amounts of amino acids. So this certainly tells us, yes, they're calling it a protein supplement. It certainly has a good amount of proteins. We are also conducting more and more of these studies. We are looking into different types of plants, both crop as well as non-crop plants, and we are trying to gather as much pollen as we can. And we are certainly going to rely on citizen scientists to be able to collect more pollen. So if you're interested in contributing pollen to our project and to help build this database, please get in touch with me after my talk or later on when you can, and I'll help discuss with you what the protocols are. And we are very soon we are going to come up with an instructional video to help share with citizens how they can collect pollen, sample it, store it, and ship it to us. So I just wanted to give you some numbers that I have. Uh, I believe I have maybe like 5 more minutes, so I'll just quickly go over these. So we are constantly testing different types of pollen for their lipid content, their protein contents, their phytosterols, amino acids, and metabolite contents. And I just wanted to quickly show you some important protein informations that we have. So almond is about 30% of crude protein content. Metaform is about 24.1%. Hybrid carrot seeds is about 13.5%. Rabbit brush is about 25%. Corbicular pear, so this is something that we trapped from honeybee colonies in a pear orchard, is about 15%. And as always, with these trapped pollen samples, we are not entirely sure how much of it is actually pure pear pollen. But there is this one study from our lab. You can look it up, Topitzhofer et al. 2018. We found that the blueberry samples that we collected by trapping them from the hives, it hardly contained any blueberry pollen. We found other weeds such as scotch broom. They were more abundant in the pollen traps compared to blueberries. But this overall sample that we tested, it had about 27% pollen. California poppy has about 28%, sunflower 17%. We also hand collected blueberry pollen. We checked two different varieties, Elliott's and Duke's. Elliott's had about 18% pollen and Duke at about 27% pollen. So we also tested pure plum pollen, which is about 30%, pure almond pollen, which is about 35%. Apple is about 39%. This is pure apple. Pure cherry is 30, sorry, pure cherry is about 36%. Pure apricot is about 38%. And the last two slides that I just wanted to show you is that I know spring is approaching fast and you're certainly thinking about what to do with your hives. And this was a cartoon that I found, and I found it so funny, which is so true. And I'm sure as a beekeeper, there are way more things than just these that you're thinking about. But I just wanted to share this with you and just tell you that we all feel your pain. And we all know that what an incredible uh, service you're providing. So, just some general recommendations in terms of nutrition and how to prep your hives. So, for example, if you're preparing your colonies for spring or summer, it may be ideal to feed bees with 1:1 sugar syrup. This is because this 1:1, so 1 part sugar to 1 part water, this mimics nectar. 2 parts sugar to 1 part water is thicker, so the bees may process it like honey and they will store it in the hive. And this 1:1 sugar syrup, which mimics nectar, will also stimulate brood production. Some beekeepers around here, they tend to use protein patties in spring because this can also help stimulate brood production. But just remember that if you're feeding these colonies protein patties too early, the colony will start to grow. So if the weather changes suddenly, if it's suddenly too cold or if it starts to rain, which is of course quite unpredictable because here in the Pacific Northwest, the weather is quite unpredictable, especially in spring. So if there is no forage available, the entire colony will suffer because now that you have provided protein patties, the brood nest has already expanded. So there are certainly more mouths to feed. But with less availability of forage, that colony will suffer. So either you continue to provide protein patties, or it is best that you wait and you feed protein when you know that pollen naturally in the foraging range will be available very soon. And also same with the syrup, it should be fed once it is warm enough for the bees to be able to break their winter cluster. And even, for example, say fly out when it's warm enough in the high 50s or so. If it is too cold, or rather, we often provide solid sugar candy, which we've seen that it works well. So with that, I would like to thank my advisor, my collaborators, colleagues, our funding agencies, our partners, our stakeholders, our B Lab students, without whom none of the work that we do are possible. And thank you, Renata, for inviting me, and thank you all for being present today to listen to some of the tidbits on bee nutrition. So if you have any questions, you can always email me at basuchat at oregonstate.edu. You can also reach out to us at the OSU Honeybee Lab. You can access my personal website or the B Lab website, or you can reach out to Ramesh at ramesh.sagili at oregonstate.edu. So if you have any questions, I will be happy to take them now. Thank you so much.